Welcome dear students, I am Professor Ronika Yousuf. Today we will discuss in detail about the canal system of Porifera. But before getting started, let's have a quick recap on the classification of the Porifera. As we already discussed that phylum Porifera is divided into three classes: class Calcaria, class Hexactinellida and class Desmospongia. Class Calcaria, these include the sponge with skeleton of calcareous spicules. Hexactinellida include glass sponges which are most symmetrical. And class Desmospongia is the largest class of sponge with brilliant coloration, skeleton formed of siliceous spicules and spongin fibers. Now let us discuss about today's topic, that is canal system of Porifera. Canal system is also called aquiferous system. It is a system of interconnected canals through which water circulates, which helps a number of metabolic activities of a sedimentary sponge. On the basis of their increasing complexity, canal system are of three types. First, asconoid canal system, second, syconoid canal system, and third, leuconoid canal system. Let us discuss about the first type of canal system, which is asconoid canal system. It is the simplest type of canal system and is characterized by number first, wall. The wall is thin and unfolded with uniform thickness. It is formed of two cellular layers: outer pinacoderm, which is also called the dermal layer, and inner choanoderm, also known as the gastral layer. Number second, ostia. Wall is perforated by a large number of minute openings called ostia or incurrent pores, except at the base. These are intracellular structures and are present within the cell and called porocytes and directly lead into the spongocoel. Number third, mesoglea. It is weakly developed and is a uniform thickness. It does not form dermal or gastral cortex. Number 4, choanocytes or collar cells form the gastral layer which lines the whole of the spongocoel. Gastral layer forms a continuous layer except when interrupted by the porocytes. Number 5 is spongocoel. It is large and spacious. It opens out by a single large and circular aperture called ossiculum or excurrent aperture, present at its upper end, and it is often guarded by an ossicular fringe of long monoaxon spicules. Asconoid canal system is found in the members of order Asconosa or Homocilla, for example, Leucosolanea, Olanethus, etc. These sponges have a vase-like body and radially symmetry. Route of water flow. The water comes outside through the dermal ostia, then it goes to the spongocoel and goes outside through the ossiculum. Second type is syconoid canal system. It is more complex than the asconoid canal system and is found in a large number of vase-like and radially symmetrical sponges. It is characterized by number first, wall. Wall is much thicker and folded due to thickening of mesoglea, which may form cortex in certain areas. Number second, dermal ostia. These either open directly in the radial canals or through the incurrent canals but never open directly into the spongocoel. Number third is radiant canal and incurrent canals. These are infolding of the dermal layer, form either incurrent space or incurrent canal, which are lined by pinacocytes, while unfolding of spongocoel produces the radial canals. Incurrent canals and radiant canals become alternatively arranged. Number 4 is choanocytes. Choanocytes are restricted in the radial canals only. Number 5 is spongocoel. It is the narrow and lined by pinacocyte. It opens out by ossiculum. Syconoid canal system is derived from sphenoid canal system by folding of body wall and thickening of mesoglea. It is confirmed by the presence of oleanther stage in the life history of cycon and confining of collar cells in the radial canals. There are 3 grades of cycloid canal system. Grade 1 is characterized by its radial canals are formed by evagination of spongiocele and are lined by collar cells. These are completely surrounded by water and are separated by wide space, which is called incurrent space, which are not externally bounded by a pore membrane. So, organized incurrent canals are absent. Their dermal ostia lead directly into the radial canals. Radial canals directly open in spongiocele by wide aperture called internal ostia. There are number of excurrent canals in grade 1. Their mesoglea is of uniform thickness, so does not form dermal or gastral cortex. Their spongiocele is reduced and narrow, and the route of water flow is water comes outside through the dermal ostia, then enters into the radial canal, then interostia, then spongiocele, then ossiculum. After that goes outside. Grade 2 of canal system. It is characteristic type of a cycloid spons of class Scalicaria and is characterized by radial canals come closer near their outer ends, so enclosing tubular passages called incurrent canals, which are formed by invagination of dermal layer, so are lined by pinacocytes, while radial canals are lined by collar cells. Incurrent canals are externally closed by porous pore membrane. In a tangential section, each incurrent canal is surrounded by 4 radial canals and vice versa. Dermal ostia are intracellular microscopic apertures in the pore membrane and are surrounded by contractile pinacocytes called myocytes, which act as sphincters. These lead into incurrent canals, which are connected to the radial canals by short and narrow intracellular canal called prosopiles. So, these are comparable but not homologous to dermal ostia of sphenoid canal system. Then mesoglea is much thickened and form gastral and dermal cortex, inner to incurrent canal and outer to radial canal respectively. The gastral cortex pushes the collar cells more outwards, so small excurrent or efferent canal is formed in which radial canals open by a wide and circular aperture, which is called apopyle, present in the center of a circular diaphragm and is regulated by myocytes. Excurrent canals open in a spongiocele by gastral ostia. Their spongiocele is reduced and narrow. It is lined by pinacocytes and open outside by a apical ossiculum. Now, route of water flow. Water comes from outside through the dermal ostia, enters in incurrent canal, then through prosopiles enters in radial canals, then through apopiles enters in excurrent canal, and then through gastral ostia it goes to spongiocele, and after that goes outside through ossiculum. Grade 3 of cycloid canal system. It is similar to grade 2 of cycloid canal system, except that mesoglea is much thick, especially the dermal cortex, due to which incurrent canals become long, narrow and branched, which may form large cortical space called hypodermal space. If you found our video on the canal system in sponge fascinating, don't miss out on the next video. Thank you and hit subscribe for more informative lessons on biology. This is Professor Ronika Yousuf signing out.